and welcome to today's webinar on BioStudies. My name is Anna Swan and I'm a Scientific Training Officer here at EBI. So if I just hand over to Ujis Sarkans, who will be the speaker for today. Thank you.
Hi everybody, so my name is Ujis Sarkans and I will talk about the BioStudies database, why it exists and what it is. So I will start with some introductory thoughts, then we'll talk about the motivation for BioStudies, and then we'll show sort of on a very practical level how to access data in BioStudies and how to submit data to BioStudies. And then we'll finish with some advanced uses, potential advanced uses for BioStudies and future ideas. Very quickly, to introduce myself, I have computer science background from the University of Latvia. That sometimes is a snowy place, but not now. Now it's summer there as well. So I've been at EBI since year 2000, so quite a long time. Currently, I run the functional genomics software development team. We develop and run the BioStudies database, but then we also maintain software for ArrayExpress, which is a gene expression database. And then we also participate in building the bioimage archive, which was announced quite recently, a couple of weeks ago. We do in collaboration with other teams. So very quickly, my team is software engineers, currently 6 of them, working across these areas, and then being involved in various software component processes, sort of. So let's very briefly talk about research data management. So it's difficult to summarize on a single slide, all the best practices for data management. But well, there are a few things that people should do to make sure that the data that they produce and share is actually understandable and reusable. So the experiments and samples, protocols, and so on need to be well described. Ideally, using existing standards, existing ontologies. The data is ideally provided in both human and machine readable format, and then the data should be submitted to appropriate databases if such databases exist. Some quality control information also should be provided, and so on. So there is much more information available in this Train Online course, bringing data to life, data management for the biomolecular sciences. So here I'm just trying to summarize very briefly and to introduce, to mention these problems in the context of why BioStudies database exists. So as a result of life sciences being essentially more and more data intensive, there are more and more databases that are used, more data is generated, and eventually it becomes difficult to track all the data associated to a paper. So let's briefly look at this paper, which is about conservation of milk utilization genes. When one reads this paper, the questions could be sort of what data have been submitted to various community databases that are relevant to this paper. Which are those databases? Are all the associations to the data sets of the same nature? And what else? What other data are attached to this paper? So in this case, I've just copied out some paragraphs from this paper. So there were some nucleotide sequences deposited in the GenBank database under accession numbers this to that. Then the paper also mentions the microarray data reported that have been deposited in the gene expression omnibus database under accession number. So that is actually the ArrayExpress database accession numbers. So there was a little bit of a mistake here. It's not a gene expression omnibus at all. So this probably does not make the data easier to find. Also, there were some primers used, and then a GenBank accession number. There's sort of some pre-existing data was used when designing an experiment. So there were also some supplemental materials produced and published, some 3 files in this case. So the BioStudies database, essentially the aim is to facilitate transparency and reproducibility of research by aggregating all the data associated to a study into what we call a data package. We have the latest publication about the BioStudies is here. So I have just one conceptual diagram that hopefully quite clearly illustrates the main components of the data package. So the data package consists of some metadata, well, who generated the data, what the keywords are, what are various dates associated to this data set, and so on. So then there are links to the components of data in various other databases, for example, PRIDE for proteomics data, metabolites for metabolomics data, and so on and so forth. And then, well, there are also data files that did not fit anywhere else, that sometimes are called orphan data, so some Excel files, images, and so on. So these are the 3 main components of a data package, and then we also link back to the publication in the Europe PMC database. So if we return now to our example paper, and if we look at this paper in the Europe PMC database, then there you will find the data tab that will link to BioStudies. So that is then the data behind the paper. And then now if we follow that link, then we find the BioStudies entry that was constructed from this paper. So there is, again, the abstract of the paper, all the contact details, and then the supplemental data files are all available here. That's the thumbnail for one of the files, but of course all the files are available for download. And then all the accession numbers in ENA and in ArrayExpress were extracted from the paper, and the links are provided from the BioStudies entry. So all this was done automatically without any involvement of the authors of this paper. This diagram, we will not spend much time on this, and but the point of this diagram is to show that there are essentially 2 types of entries in the BioStudies database: those that were created post-publication automatically, in this case ingesting data from Europe PMC, and data sets that were prepared actively before the paper was published. And I will have on another slide some data sources on the left-hand side as well. So what are some of the problems with automated construction of data packages? This automated process that created the BioStudies entry could not distinguish between the data that was generated as a part of this study and the data used in the study as a reference. So remember, there was a paragraph about primer use. Also, the paper actually mentions a range of accession numbers, while the automated process could only find two of these, so the beginning and the end of the interval. So that's another problem with an automated approach. So optimally, the data packages are created before the publication. In that case, links from the paper to the data package can be created, and in general we have the community has a better opportunity to have better structured supplementary materials and data. So returning to this diagram of various data sources for BioStudies, again, I will not spend time on explaining all the boxes here, but manual BioStudies deposition is possible, as we will see a bit later. Okay, so I think that's enough motivation for the BioStudies. So let's look at how to access data in BioStudies. So if you go to EBI/BioStudies and then click on Browse, you will see that currently we have 1.2 million data sets in the database. Most of those were imported from Europe PMC automatically, but there is also a growing number of pre-publication data submissions. You can see the various data sources on the left. So besides Europe PMC, there are several other data sources, and I will talk about some of them in a minute. There are other facets that users can use to, for example, find the data from a particular year and so on. And then here on the right-hand side, the data sets can be sorted by release date or by some other attributes. A general note here is that, well, usually we think that a data package will be found coming from the paper. So probably it's not worthwhile to build a very complicated data exploration interface for BioStudies. Well, still we have done some work on this so that the individual data sets also could be located looking at the data set space rather than at the paper space. Scrolling down, looking at other types of facets that are available in BioStudies. So there are, it's possible to find, for example, only the data sets that include protein database links, or only the data sets that include images as TIFF files, and so on. So now looking at a single dataset representation. So this is a large imaging dataset in BioStudies. Again, on the left-hand side, you can see study annotation, and on the right-hand side, the files and the links. In this case, this particular dataset has 300,000 files. So we have not itemized each and every file in this view. There is just an index of all the files. And then the individual files are available from this FTP location here. And there are also some other external links for other views of this particular data set. Another example, we have a collaboration with the Source Data Project. The idea of the Source Data Project is to make available not only the figures as sets of pixels in PDF files, but also the data that was used to generate those figures, as well as the various entities that are present on figures, such as genes or compounds, have been annotated, and so that's really all about making the figures richer and easier to use in subsequent research. So in this case, well, again, we have study annotation, of course, but then the annotations of the biological entities are available as files here on the BioStudies page. All these attributes are also searchable, so you would find this particular data set if you would search in BioStudies by a particular gene or particular molecule. And then the data files are, in this case, Excel files. They are again available, as well as the links to source data site for the actual figures. So now queries. It's of course possible to query for data in BioStudies. If you type in gut in the search box, you would find almost 15,000 data sets. A search is ontology enabled. That means that a search would also find data sets that mention more specific terms, like in this example, or synonyms like digestive system. How does this system know about synonyms and other relations? So we rely on the experimental factor ontology, which is another project at EBI that aims to provide a systematic description of experimental variables. For example, biological sample characteristics, and so on. So please go to EFO to find more information. Now back to BioStudies. So it's also possible to combine search terms. You can use and, or, not, and brackets. So in this case, we are trying to find all the data sets associated to lamprey gut. We are finding 9 results, and also the ontology expansion still works also in this case, as you can see here. It's possible to use brackets to group search terms, leukemia and either mouse or human. Then you would use a query like that, or can be skipped. So that is a default. If you would just type in mouse space human, the outcome should be the same. And then again, because of the ontology enabled search, Homo sapiens could be used instead of human. As in this case, it should be then put in quotes. Finally, talking about the data access, it's also possible to look at all the data sets associated to a single project only. So if we look at only the data sets imported from Europe PMC, in addition to the facets that you already saw earlier, you would see the funding agency facet. So all the data sets coming from Europe PMC would have a funding information attached. So and that information then can be used to narrow down your search. So that's all about data access. We hope that it is easy to use and intuitive. If you have any suggestions how to improve that further, we are very happy to listen to any feedback. Now, how the data submissions would work. So we have a data submission tool that, first of all, it's flexible. So, well, because BioStudies is so generic, we need to be able to deal with various types of annotations associated to the whole data sets or to individual files within those data sets. We provide both a web form as well as a tab delimited file for file upload mechanism for submitting data to BioStudies. And we also provide configurable templates for different domains and projects. So that the last functionality has not yet been deployed to the production instance, but very soon you will be able to see a different template for submitting imaging data sets from any other data set. Just looking at a few screenshots, well, the registration screen is hopefully self-explanatory. Just to note that we also encourage adding ORCID identifiers to your user login. Then once you have logged in, you would see the And after already submitted a few data sets, you would see a screen like that. So that is a list of your submitted data sets, and this other tab would show you that the data sets that were not yet submitted, but that you are working on. So you can work on parallel on several submissions. That's not a problem. So this view is available from the submissions button here on the top right. Then the button next to submissions is files, which is really a list of files data files that you have uploaded to your BioStudies home directory. So this is really just file storage. The fact that you have uploaded a set of files does not mean that they are available through BioStudies data access user interface. So you really need to finish one or more submissions and associate files to submissions. Only then those files are available. We also have for the cases when there are too many files to upload through the web forms, we provide FTP uploads as well as Aspera. So for those of you who do not know Aspera, so that is another very fast data transfer protocol. So if you click on FTP/Aspera button, then you would see instructions like this, what you need to do to be able to submit, to upload data files using these mechanisms. And then how the screens would look like when you submit a single study. So here on the left, you would see validation messages. So these are the things that should be fixed before a data set can be submitted. For example, a title really should not be blank. And then the other tab here is about adding new elements to your data set. And then in the main area of the screen, you would see title, the public release date, description, and then scrolling down, contact information, and then links and files. So these are the main things that you package in a BioStudies data set. So for files, you will have an opportunity to select files from the list of files that you have uploaded in your Files tab. And individual files also can be annotated if necessary. Well, you can add description, but you can add your own columns if necessary to distinguish between different files in your data set. And to help with entering links in your data set, we have an integration that uses the services of identifiers.org. So if you would start typing ArrayExpress, then the autocomplete would help you to fill in that information, and the system would also know what is a valid pattern for an ArrayExpress accession number. If you do not know what identifiers.org is, please go to this URL. So that is a project run also at EBI. So that is an identifier resolution system, essentially. Finally, on the data submissions topic, it's also possible to describe your data set in a tab delimited, for example, Excel file. And that mechanism is useful if you have many similarly structured data sets, either ready at once or perhaps you might have over time similar data sets. Well, to use this functionality, you would need to learn a little bit about the format that we use. The description is available online. We hope this is quite an easy data format to use, and it should be quite intuitive. So that concludes the submission section. And finally, let's talk a little bit about other uses, potential uses of biostudies and what is coming in the near future. So data sharing in a project. So that is the case when you have a project running, you do not want to immediately publish the data, but still biostudies can be used for sharing data within a project, and then easy data publishing in future when you write a paper and then need to release the data to general scientific community. So it's possible to set up a new project. It's possible to design useful data and metadata forms and manage access rights. Well, currently this cannot be done without involving us, the BioStudies team. So you would not be able to access that functionality directly through web interfaces. But so the main point I want to make here is that for the data types where the data formats do not exist, where standard data formats do not exist, it is possible to design sort of your own data sharing approach by figuring out what attributes you need to use to describe a particular data set. How would you describe individual files within a data set? Do you need to link to information that exists somewhere else outside the biostudies? And it's also possible to create hierarchies in biostudies. And then talking about access rights management in this case. So if you have biostudies project data, then it's possible to set up individual accounts where associated to that particular project, where each individual account can upload a new data set or edit or delete their own data sets, as well as read all the data sets in the project. And then, well, project might want to set up a read-only account where people can log in and access all the data available in the project so far. And then it's a very simple process. Then once a corresponding paper is published, to make a data set public for everybody. So the second sort of future work idea is migrating ArrayExpress to BioStudies. So ArrayExpress is gene expression database that we run at EBI. If you would, if you know about ArrayExpress, so currently one of the features of ArrayExpress is, for example, to show how different samples in the experiment, what they were, how they were treated, and what the associated data files are. We are working to migrate all the ArrayExpress data to BioStudies, and this screen shows how an ArrayExpress data set would look like when it's available through BioStudies. So again, all the files, as usual in BioStudies, are available from the right-hand side. But then here on the left-hand side, now we have more freedom to design various views on ArrayExpress data. For example, we can summarize what different sample characteristic combinations were used in this particular experiment, how many samples were used for each of the combinations, and so on and so forth. Finally, the last few slides here, we can also accept a light microscopy imaging data in BioStudies. Just to briefly explain what we are aiming for here. So if we look at the paper from Cellular Systems Journal, so that paper would include several figures like that with panels of images. Typically, in a paper that involves imaging, you would show in a paper, well, maybe 20 or 30 different panels of images. Well, if it's well, the community often would like to see more, would like to have all the data associated to this particular publication. In this case, we have all the data associated to this paper. There are 100,000 images. So essentially, what then we are enabling users to do is, so if there are, for example, let's look at this here. So there may be more than 2 treatments used in this study. There may be 20 different treatments, and there may be, for example, 20 or 30 different fields of view. So having a complete data set in BioStudies, as well as BioStudies giving users the functionality to explore that full data set and essentially walking along these 2 dimensions. So that is something that we are aiming for. And in fact, well, we already have some functionality in place in this particular case. So we have 92,000 images. The user has queried by a particular cellular phenotype. So the user wanted to find all the images associated to large nuclei. So then we are showing only, well, 12,000 images in this case. And then it's possible to further narrow down your search and find only the images that you are really interested in, and then download them, for example, for further analysis. So this not all of this functionality has been fully rolled out yet, but it is coming very soon. So that concludes my presentation. Please get in touch with us using this BioStudies@EBI email, and I am very happy to answer any questions. So there is a question about databases containing virus-based data. Well, I do not have experience specifically about viruses. So from the BioStudies point of view, if well, of course, please feel free to search in BioStudies for data sets related to viruses. Let us know if you cannot find well, what you are looking for. We may be able to help with finding the information. So that's on the data access side. For data submissions, if there are no dedicated data repositories for data that you would like to share with the scientific community, then please come to BioStudies and submit data to BioStudies, and the data will be published and available for everybody. I hope that at least partially answers your question.
So we will leave it there this time. Yeah. Great, thank you.
Thanks.